pitch for why you should adopt the FAIR principles. So on the one hand, I think you've been told that you should by funders or colleagues, or I mean, you've heard that this is something that's on the move and you're supposed to be knowledgeable about what these principles mean. But I want to twist it also in the other way. So there's some intrinsic value also in adopting the FAIR principles. And that is kind of making stuff work better, integrate more smoothly together. And that's why the title of the talk is "It Just Works with My Software," which I don't really think that it's the current state of affairs, but I think it's the direction we should be moving. And I think you could, for your own research and with your own colleagues, you can actually come to a state where you can have an ecosystem of software and data and other resources that just connect and work well together. So that's a little bit of the kind of disclaimer for the talk. The FAIR principles, it's an acronym for findable, accessible, interoperable, and reusable. But it's not just those words. It also contains some encouraging directions that you're supposed to go to get your data not only shared for your colleagues, but also shared in a way that it can be leveraged by software. So it should provide some additional structure beyond just having free text. And then in addition to all of these regular incentives around sharing data in general, getting more value out of the research that the funders are investing in. And then they have requirements on this to be something that you need to do as a researcher, and then all the way to kind of this actually contributing to better research, which you can do with just having the data being shared at all. And then focusing a little bit on how would we do this more effectively by using software. I've also highlighted the kind of usable part of reusable here. So by implementing these principles, you also make the data more usable for yourself during your research. And I think the first time you approach the FAIR principles, there might be a lot of new words and new ways of working that you need to familiarize yourself with. But over time, I think as you have established some practices in your own projects and perhaps in some playground testing experiences, then you will have something that you can kind of just adopt as a template for the next run. So starting off here, I think is a general thought about how do you assess or evaluate data that has been shared in general. And in this case, you could argue that you should be able to say that the data should be readable for machines, and by machines here we mean software, without needing some specialized algorithms, heavily licensed or expensive software. Or building your own software more or less using ad hoc algorithms or something like that to get the data into a format that you can make sense of it. So that's basic agreement with your colleagues that if someone leaves, you would be able to take the data they've been working on and make sense of it. So that's the bare minimum, I think, and that's part of the good data management practices overall. At the next step here, if you kind of take the outside perspective and look at the data that you've produced, then you might need to provide some extra documentation about the process and about the data themselves for someone else to be able to feel comfortable in citing or reusing the data for another project. And that could be also yourself, your future self, looking back at the previous study, or maybe some work that was never published. And if you would like to then include it in a recent study, what would you need in terms of documentations for that to work? So, I mean, both these quotes comes from some guiding principle documents on data sharing and making that work more generally. And then if we look at this from an even broader perspective and making sure that, I mean, this ecosystem of data supporting future research and I mean, providing all these values that you saw in the first picture, then we need some common standards and ways of working to be able to know that if data has been shared by someone in this field, then I can trust that they should have used one of the formats that we usually use for a certain type of data, or that they've described the information using the terms that we need to make an informed assessment from them. These numbers, I think, are a bit old, but there are registries now that kind of try to survey the terrain for what are the different formats that have been used in certain fields, and the same for terminologies and guidelines. And if you're a new researcher in a certain field, then I think a first step could be to explore, so what are the available standards and formats that people are using? Or, I mean, if you are feeling that something is not really working as it should be, then this could be a nice resource to start exploring. And if some standards are missing, I think there might also be some incentives for more mature or I mean, researchers who have been active for a while to start a movement on maybe creating some common guidelines or terminologies that would enable wider data sharing in their community or for their specific type of research. This also leads into other principles that are related but not exactly the same. So we have the FAIR principles here in the middle. So that's the kind of findable, accessible, interoperable, reusable, and not necessarily open, but ideally as open as possible, given the possible restrictions for, I mean, if it's human data or if it's sensitive for other reasons like copyright, then you can still apply the FAIR principles. But then you also have the idea that this should not be only for people, it should be for machines. So the idea is to make it more and more easily integratable. Then on the other end, you have open science, which pushes for more parts of the research process to be opened up and sharing different types of outputs, not only the data, but maybe also the code that supports some of the analysis. It could be some of the protocols for the wet work. I mean, anything that would make sense and enrich the research community really should ideally be shared. And perhaps even data of failed studies to, I mean, make sure that people don't replicate or do the same thing unnecessarily. You also have reproducibility, which is also increasingly a requirement from funders, that you provide the proper documentation and sometimes even the actual code that supported the generation of some parts or all of the results. So, I mean, being able to take the same data and the same software algorithm or configuration, and then see that you can actually get the same results from a computational process or an experiment. That's something that increases trust, but can also be a useful rubric for yourself when you're planning to share your work with your colleagues. So, I mean, you could build in both of those directions, but here we're going to focus on the one in the middle, the FAIR principles. I think you can consider this as a spectrum. So going from this, I mean, if you've done something once, you've got the results, and then you just throw the data away, I mean, that's usually not acceptable if it's something that you would use in a published context. But sometimes it's fine if you end up with, I mean, doing an analysis and you don't really get the results, if it's just an explorative thing and you see no potential reuse value, then you might be on the lower end of this. And then the other extreme is that the data should be described and organized in a way that basically makes it reusable for every possible use case, both currently, but then also in the future. So, I mean, these two extremes, somewhere in the middle is probably where you're going to end up. I mean, you might have a benchmark for now. I mean, given some of the extrinsic requirements that you need to be fair under certain conditions or for a certain grant or something like that. So identifying like there is a spectrum that you can walk across, and you can incrementally do more to become more open or more reproducible or more fair in this context. The FAIR principles, I chose to illustrate them with a search bar, this collection of different types of specifications or standards, and then this graph or network of different connected outputs. So we could imagine that the idea of the FAIR principles is building this network of linked data sets to publications, to protocols, to code, and whatever is interesting for a researcher to be able to navigate and explore. And ideally, you should be able to download all of that perhaps as a package in some standardized piece of software. And the search box here, that indicates that there might be some keywords or terms that you would expect to be able to browse through or search using. So, I mean, depending on what type of research output it is, often in the life sciences, you would be interested in, for example, which organism is a certain data set or specimen related to. And then there might be then a taxonomy that you could use to help you browse or reduce the number of hits in a hit list. And then the formats and other kind of terminologies, ways of saving or recording the information, that's just making it easier for you to then download it and make sure that you can integrate it with the other data that you're working with. So if you then write a query, get a long list of results, and then you basically put those in a shopping cart, download them. And if the formats and terminologies are, and the stars perhaps are well aligned, then you will be able to integrate these with minimal effort. So moving towards a web of fair data and services, like the internet kind of revolutionized how we approach information. And I wrote here from Gutenberg to Berners-Lee, which is a reference to the inventor of the person who popularized the printing press, Gutenberg, and Berners-Lee, who conceptualized and was a big proponent or had a big stake in making the internet come to reality. And I think there are a few pretty down-to-earth questions that you can use to guide yourself through the implementation of the FAIR principles. And the first one here would be, so what kind of research output are we really looking at to start with? So, I mean, is it a DNA sequence, or is it some other kind of material that the data represents? And what would be the appropriate way to share that information? And what are the practices related to that in the community that would be interested? Where will people be looking for this? And that would indicate which database you should submit it to, or if there's a registry where the information should be indexed. And then who should be able to get access to this data? So if there's any restrictions related to, I mean, the copyright or if it's sensitive data or something like that, as I mentioned before, then this is something that needs to be made clear. And if there are some standards or specifications that are customary for this data type or discipline or type of experiment. And then I put what else will be expected by the research community as kind of a catch-all for the kind of rich descriptions, documentation, or metadata, as we will be calling it here, that will be needed in addition to the data files in a standard format. I will present this kind of what types of research outputs we're talking about as one of the prerequisites. And this isn't really specific to the FAIR principles. This is basic good data management practices. So, I mean, if you look at your research project from a kind of start or planning to communicating results timeline, you might be able to identify some things that you should be recording or saving some notes on to be able to then later on share the data and for someone else to be able to assess if the process is sound, and also perhaps making sure that the important information is actually being saved in a file format that's appropriate or easy enough to then just upload somewhere in the appropriate databases. Then we have basic data organization practices. So, I mean, for your own project locally and with your colleagues, finding a good way of organizing this information. So you can see, so how do the different files that we've produced relate to this documentation that we've accrued over time, so you can get back to it. And structuring this in a effective way that ideally should work with the software ecosystem that you've built would then make it a lot easier to take the next step and adopt the FAIR principles. So starting to think about findability, accessibility, and so forth. You should also kind of, or you could also go the other way around, of course. So looking at what are the common practices for the different principles, and then look at, so what would we need to include in our prerequisites when we look at different outputs? What should we be describing and documenting there? The first recommendation then for implementing the FAIR principles is actually to start with well-managed data. And we have a resource at SciLifeLab, which we call the data guidelines, or SciLifeLab data guidelines. And I'm going to jump into these to give an example of how you can navigate this information resource to help yourself implement the FAIR principles. So I'm going to switch windows for now, and let me see. So this is going to be a normal browser window. And I think this is the one. I hope the text is large enough. I see a thumbs up. Yes. Perfect. So these are the RDM guidelines. The start page looks like this. And if you click, for example, share, which is, I mean, the context for the FAIR principles, sharing data, then you can see some general guidelines here on, I mean, how you would find a suitable repository, depending on the type of data you're working on. And I think this is actually a good starting point also for adopting the FAIR principles. So looking for, are there existing repositories that you should consider depositing your data to, and see if they have some recommendations or some solutions that help you implement the FAIR principles. So this is a good starting page in general. There's also a link here to a topic for the FAIR principles more specifically. And I think I've already opened that one here. So here, the actual principles as they were presented in the original paper from 2016, they are listed here with some explanation and proposed actions. And I mean, digesting this might not be super easy. These are articulated in somewhat of technical jargon. And there is then a supporting translation of the principles into more common language linked from the top of this page that I think will serve as kind of a good starting point. And I'm going to zoom in here on one example. So let's see. And the first principle under findability here is that metadata are assigned a globally unique and persistent identifier. And an identifier in this case is something like a digital object identifier, a DOI. It could also be an accession number from a database, in the life sciences. And the purpose is that it will allow you to cite the data set, and then over time, in the far future, you will still be able to use that identifier to get to the data set and download it. So, that's one key thing that you should provide for implementing the FAIR principles. And then they also have a description of the researcher's responsibilities here, and something that you might want to require from the repository where you would be depositing your data. So some of the technical aspects here, like issuing this persistent identifier or globally unique identifier, that would normally be something that the database or a repository does on your behalf. If it doesn't, then you might consider, so are there any other alternatives then or approaches that I could adopt to get this identifier? But ideally, this would be solved, and I think that is the case for most of the databases that we recommend in the RDM guidelines for different types of research. There are some other principles here, like each that the data should be described using rich metadata. And this word metadata is something that recurs in many of the principles going forward as well. So throughout all of the principles, and it basically means that you should have some form of structured documentation of the data that gives you information on how it can be processed, how it was created, and can serve then as some sort of filtering criteria when you search for it. So, if you can find it using its identifier on the one hand as part of a citation in a paper, then you should also be able to browse and find the data set using keywords or other properties that are structured in the database. So you have two ways of getting to it. And then there are some other parts of the principles here, but I leave that as an exercise to you to browse through the principles here in the RDM guidelines. And I'm going to pop into an example of an actual paper. So this is a paper published in the Cell, let's see, and we have data and code availability here. And I'm going to make this a little bit larger. It's not like you're going to need to read the paper, but still there's then a heading here for data and code availability. And then they have a link to a database called ArrayExpress. So this is a database for functional genomics. And if we click on this one, we will end up in the ArrayExpress database. So we have a link then that serves as a identifier, and we can also use this accession number, which is a shorter form of the URL to get to this specific data set. And here you can see that there's a lot of information about the data that could be used for browsing the database as well. So you have the type of organism, which is linked to a taxonomy. And then there are some descriptions of the different samples and the type of experiment and some other technical aspects that might be interesting to someone looking for similar data sets or being inspired by something that someone has done before to do something similar. You could be interested in the way they set up the study, reusing some of the protocols without necessarily reusing the actual data in this case. Let's say then that we were in a situation that we had functional genomics data, and we were looking at this database as a potential venue where we would submit our data to as a first step of making it fair. Then for this database, we can see that there are there's a submission guide, and there's a description here on some of the general guidelines that they promote or are in compliance with. So I'm not going to go into detail on these, but these are guidelines that have been produced in a large community over a long time to describe sequencing experiments and other types of experiments. So some minimal reporting standards, you can call them. And then if you look at the submission guide, then there are detailed descriptions on what you need to prepare in terms of metadata. And that would be, for example, so which characteristics of this data do you need to include? And I mean, what kind of detail would be expected in the values? And then if you record dates, for example, which format should the dates be recorded? And some of that might then be regulated in the metadata reporting standards that are proposed. There's also some recommendations on file formats here. And I think we will revisit that in a few tabs. So I'm going to switch over to the next one, which is accessible here. And I'm going to gloss over this a little bit. But I think if we look at the choosing a repository rubric here, then I mean the first choice is basically does this data set that you have contain any restricted information that cannot be shared in one of the public databases? And then you will see that the number of options will decrease quite dramatically. So one database that we highlight here, and that this is not the only one, is that you can submit human data to this federated European Genome Phenome Archive that we host at NBIS. If it's restricted for other reasons, then there might be other databases that are more appropriate. Then you have discipline-specific data repositories, and we recommend that you use them if there is a discipline-specific or data type-specific database for your type of experiment. And then they will have pretty specific guidance on which file formats and metadata checklists you should start from, which makes life a lot easier. If there's no specific database for the type of data, then you will need to use more of a generic database. And then you need to do some work on trying to figure out, so which are the standards that would be appropriate? Which documentation should I provide? And I mean, if there is a lack of standards, then maybe your community needs to do some work to actually develop some common agreements on what good sharing practices are. And the most general version here is to have these databases that are not associated with your group specifically, but can serve any community and just contain any data type, just like the institutional repositories. So with that, I think I'm going to jump over to the examples from the RDM guidelines. So here you can, under the sharing phase, find some recommendations. I mean, this is the first page I showed. If you scroll down a bit, you will find these recommendations for discipline-specific repositories. So if you're working on genomic data, there are some examples here, and then descriptions on when you would use it and some of the guidance related to those databases. So this is a good starting point. And then if you go to the interoperable part of the principles, then these also cover the file formats, the ontologies or taxonomies or controlled vocabularies, the way you express your data values and maybe the column headers, for example, in your tabular spreadsheets. They should be standardized to the degree it's possible. And in this case, they use the word formal language for this. So it's basically meaning that there should be some sort of common way of expressing the information that is easy to integrate with other, ideally other disciplines. But I think practically you should start with, I mean, your research group, making sure that you have a common terminology that you're working with, then your discipline, making sure that the data that you submit to the databases are easy to integrate. And when you share and work together with collaborators, then there's minimal chance of getting some confusing or conflicting definitions of the same terms, and having your pipeline struggle with getting the data files to ingest properly. Here we have also a section in the RDM guidelines that specifically covers metadata, and that also then includes a section on these controlled vocabularies and taxonomies and so forth, and how they can be used to create documentation or structured documentation on the tabular data and other kinds of data that you have collected. It also contains some links to useful resources where you can find more of these guidelines, and this ontology lookup service that can be used to find ontologies or a specific term that you might not have a standard definition of. And reusing terms is most of the time better than trying to come up with your own definitions. Because the likelihood of someone having reused a definition from somewhere else is higher than they coming up with the same definition as you, that you did in your group on your own. And this also contains some references to file format standards that are relevant for different types of experiments. So just the data dictionary that I mentioned briefly here. This is a brief overview of what this means. So it's basically just a list or a table that describes another table, where you would have the variable in one of the columns, the variable names that you actually use then in the actual data set that you're describing using this data dictionary, and then some of the properties of the variable and how it's been recorded. So for example, the unit of measurement, and I mean, if you have some ranges of expected values, or if it's a restricted list of certain terms, then which terms are allowed and expected to be in your data set and so forth. So I think this is also a useful resource to look at, and overlaps between both the FAIR principles and the good data management practices in general. We also have a section here on, I already mentioned the data file formats. I think I maybe went ahead a little bit before my planned walkthrough. But I mean, a combination then of the metadata standards, the ontologies that you're using to record information that you collect in a tabular format, and things that might come from your instruments and be generated in a sense from pipelines that come from external suppliers, or is more of a kind of recording of information that's being captured by a sensor or processed, post-processed from a sensor reading. And then there's things that are in between. So for example, the BCF file format, and also the SAM, BAM, and CRAM file formats, then you can embed metadata inside of these files as well. So then you might also want to consider, I mean, if you have the possibility to influence what information gets embedded in those files, how would you do that? And are there situations when you would use the embedded information in your internal workflows or with your research community? This is an example of the data set that I referenced in the paper that I referenced in the beginning. So this is the actual type of experiment setup. So we have a single cell experiment where you have two different samples being derived from different parts of the organism in different wet work processes. And then there are some standard definitions of types of steps in this process, or protocols for certain types of protocols that you could describe for this process. And then there are also some categories of information that you're expected to collect for a submission to the ArrayExpress functional genomics database. And then they also have some recommended file formats that you can upload, and you need to collect all of this information to do the submission. And you usually have all of it, but it might be good to prepare early, so you make sure that when you start kind of collecting all the PDF files and maybe copy pasting some of the information from emails and so forth, that you do it in a format that makes it easier to do the submission and make your data as fair as possible already from the beginning. And this is also an example of how you can describe the samples themselves. I mean, there will be many different recommendations depending on what kind of experiment you've done or the type of organism that you're looking at. So these are some examples that were relevant for this one from one of the generic sample checklists in the European Nucleotide Archive. And here you can use ontologies to define some of the terms, so it enables browsing and searching down the line. And then you have reusable here, and I think I've covered much of the rich metadata already and the motivation for that. But one thing that's I think comes in as a special thing under reusable here is the license. So you make sure that, I mean, you look at under which conditions the data can be reused, and ideally try to choose a license that is as permissive as possible. So you get the acknowledgement, but maybe not put too much restriction on the integration in other research down the line. And of course, I mean, everything should be according to the community standards. And I think the repositories is the entry point, ideal entry point for that. But sometimes you can also get some guidance from the journals. And in this case, they have something called STAR methods, which includes a key resources table that follows a certain structure of how you could describe the different parts that were used in preparing the study. And I mean, if there were any kind of concerns with certain batches or certain kits being used for different steps in the process, then it would at least be clear here what happened. And you can also find some identifiers here to certain, I think, could this be a cell line or something like that? I'm not sure, to be honest, but you can use these identifiers to identify other things than data. It could be physical things, it could be things that are used as kind of references or standards themselves as well. So that's good to keep in mind. And with that, I think I'm gonna just wrap up with repeating some of the recommendations now with some slides as well. So we have, and let's see, now I should be sharing the same screen as before with the slides. Good. And then we have the first tip here, which was start with well-managed data. And the data guidelines is a good starting point here. And then finding a repository that fits your data is the second tip. And then the third tip is to, I mean, just keep your research well organized for yourself and your colleagues. And I mean, the more you can structure that and find a reusable template for these processes, the better. And then the fourth kind of builds on top of it. So you should document your approach. That includes the data organization practices, and then also how that ties into the FAIR implementation or how you've approached the FAIR principles. And this has a second aspect to it. So think of this as kind of an incremental improvement. So building your own and improving your template for future occasions. And the final takeaway message is that, I mean, going back to the scale from use it once and then throw away for the data and then usable for everything in the future, then you might have started at some base level, and the next time you might have some time and resources and have learned something, so you can take an incremental step and become a little bit more fair. And with that, I'm gonna stop, and we can take some questions.